What I would like to talk about today is synthetic biology in a plant context. And often when we talk about synthetic biology, the first thing that comes to mind is this idea that you can use DNA as parts. The idea that you can, for example, have simple encoded features or functions incorporated into parts that can be assembled into devices and circuits and systems and so on, and implemented in living systems in at least what we hope will be a more rational process for both harnessing the information within these lower level DNA parts and to establish better engineering principles for putting these things together. And this process of taking parts and stringing them together, of course, is one thing, but the key issue is that you get the benefits of abstraction and decoupling with this process. These are features that are found in all forms of human engineering, where when I say abstraction, it means that parts that might be defined as have a particular feature means that people can use those parts without knowing underlying detail. They're shielded from underlying detail. And this process of abstraction also allows decoupling. So if you're designing a circuit, the decoupling of the underlying details of the part mean that the fabrication side of it is enhanced in the sense that you can have large numbers of people working together, and you now have a layered structure where people don't have to know everything from top to bottom of the system, but can work within a framework and deal with much more complicated structures and systems and engineer in a sensible way. And of course, this is the kind of engineering principle that we find throughout mechanical engineering, electrical engineering, chemical engineering, where these levels of abstraction are absolutely key. And of course, this is why synthetic biology is what it is. This is why we're here talking today and why the field is expanding. It's something like 30 to 50% a year with this huge levels of investment going into these new engineering principles in biology. Because many of the problems that face us over the next century are all about sustainability, all about using biological resources in better ways. And that means engineering. But what I want to talk about today is sort of incorporate some of this nuts and bolts aspect, but really it's a bigger picture. And I've borrowed this image or time-lapse movie from the BBC Natural History Unit in Bristol. This is one of the David Attenborough programs. And what you can see up here is there's a leaf-like structure. You see this stem-like or stolon emerging from the tip of the leaf. And you can see there's this nub of tissue at the end of the stolon. And it reaches down, so unlike most plants, it's going down, not up. And it's growing down for a purpose. It reaches the ground, and this nub of tissue now expands, and you can see it expanding by virtue of cell division and expansion and differentiation to make a pitcher of a pitcher plant. And of course, it's resting on the ground because it's going to have this cap-like structure here, which will pop open, so it's a hollow structure, and then it will fill with water and digestive enzymes, and it will fulfill its role as a supplement for things like phosphorus and nitrogen, capturing insects and the like in the pitcher plant. And so effectively, if you're thinking of it from an engineering perspective, what we've just viewed here is the assembly of a structure, an engineered structure by a biological process, of course, by local cellular processes. And what we see here is effectively a coffee cup. We've got the vessel itself, cap, for example, over the top of it. And in many instances, in detail, it's actually quite close to a coffee cup. If you look at, for example, at the top lip of a pitcher plant, you can see it's got this a curved rim. It has a waxy epicuticular surface on the inside of the pitcher, which acts as both waterproofing, but also you can see this lip here where insects that fall in can't find their way out again because they're trapped here by this lip. When you look at your average disposable coffee cup, you've also got similar kind of features where you've got the curved lip, which is obviously to prevent spills rather than having anything to do with insects. And the inside of the cup is lined with polyethylene to stop leakage. And so we've got this two engineered structures, which are quite homologous in a way, or similar in function, but they couldn't have been made in two more different ways. If you think of how the coffee cup is made, it's made by harvesting natural resources, stamping out templates in automated processes being assembled by the billion. There's something like 200 billion disposable plastic cups, paper cups made every year on the planet, 200 billion. And they're all made pretty much the same way. They're made as a template and stamped together. Whereas the pitcher plant version of the coffee cup comes from a single cell originally by virtue of local proliferation, organization, and there's a self-organization and emergent process at the root of this pitcher plant structure, which is both the challenge and the prize. It's a challenge because if we don't understand how that works, except in broad general ways, if we want to engineer that kind of structure, we have to understand it, we have to be able to manipulate individual steps, which themselves are more akin to population-based self-organization rather than the process of stamping out a coffee cup in a production line. And the prize, of course, is that if you understand those processes, you've got this huge benefit of being able to create material by self-organization where the resources required and the information required is intrinsic, is built into the system rather than imposed from outside. And that will come back as a theme through the talk. And so if we use the pitcher plant as a paradigm, or at least an ideal of what the kind of things that we might want to engineer in a multicellular system like a plant. Well, of course, this cartoon shows what is at the root of that process. So we have individual cells, and this is a cell that's expanded and undergone DNA replication, undergoes cytokinesis, and produces two daughter cells. And I use this image to emphasize that the process of cell division and production of daughter cells is quite distinct in plants compared to animal systems. Plant cells have this semi-rigid cell wall that encloses each cell. As it undergoes cytokinesis and division, a new cell wall sitting between the two daughter nuclei here is built internally. It's like subdividing a room. It physically builds that new semi-rigid framework or partition inside the existing cell. So those two daughter cells and all of their descendants are always going to be in the same relative position, because plant cells, unlike animal systems, there's no cell migration. The cells are locked together in place, and those two cells and their daughters will form this clonal boundary at precisely that topological barrier. And so this process of the physical aspect of growth and the consequences of growth are quite distinct. It makes it very easy in plant systems to track the process of cell division or lineage of division. And it also means that the physics of growth are very different in plant systems. And if you think about, now sort of abstracting this process, if you imagine a single cell inside that pitcher plant, which may end up with billions of cells, but if any particular cell at a given time may be triggered to grow and divide, and so we schematically show this as the cell undergoing division in time to produce two daughter cells. And we know from many years of plant development of biology that the behavior of individual cells within a plant tissue is largely determined by local interactions. There's local cell interactions. Neighboring cells are exchanging information in a two-way or multiple-way relationship, and that information exchange determines the behavior of those cells largely in a reciprocal way. And so we end up with two daughter cells who have been triggered to divide and be positioned this way in the plant organ. They can't move away from each other. They're stuck next to each other, but they're all by definition in different positions. So they have different neighbors. They can then build on this small asymmetry and amplify that asymmetry to create larger and larger differences across the tissue, or maintain them for that matter. So you've got this process of gene expression in lockstep with the microanatomy of growth, the physics of growth. And I've tried to represent that in this way here. So we've got, for example, an individual cell here which has local subcellular properties, has a cytoskeleton, has a polarity, for example, has a particular property which within the larger tissue, it of course is part of a network of cells which are close to each other, which can be exchanging genetic interactions. So each cell will have a particular genetic state at a given time. Along with its neighbors, it will be in a position to exchange information and to create a self-reinforcing network of genetic interactions. And those genetic interactions can, of course, trigger different patterns of cell division and differentiation, and so you end up with tissue-like arrangements in this rather abbreviated version of what goes on. And you end up with tissues that, of course, have physical properties based on the internal behavior of cells inside those cohorts. So the number and arrangement of cells that are dividing, for example, or expanding, create different physical forces across the tissue, which then in turn can feed back on individual cells within that construct. And we know, for example, that the size and shape of cells has a direct relationship to forces that play out across the tissue, either on the geometry and in some cases the tension or stress across the systems as well. So that's a slightly controversial area, but we do know that physics plays an important role in the behavior of individual cells in this process. So we have this, you see, sitting on a loop here with arrows because there's a feedback between all these processes. And it gets back to this issue of how material is growing and how the internal logic of gene expression plays out in the physical world. How do you go from a single cell or a small group of cells to create something which is relatively precise and well organized as a pitcher plant without this kind of interplay between genetics and cellularity? And so that, I'm sort of hammering away at this because this really underpins our entire approach. So the idea that if we want to really understand this kind of interlocked approach between the interlocking between gene expression and cellularity in physics, then we need a simple plant chassis. We need something that's easy to culture and work with, streamlined genome preferably, with exposed forward development, so we can visualize directly what's happening as natural processes take place and as we manipulate it. We need open systems if we want to, in the long term, address these kind of problems. We're talking effectively about reprogramming multicellular systems via reprogramming cellular interactions. We need to have larger scale genetic circuits and access to reliable parts. And the visualization of those processes, so we're not just looking at gene expression, we're looking at it in the context of the growing physical plant. So we need something like that. And of course, those of you who are familiar with plants will know that in higher plants, all of those things that I just said are quite difficult. You've got long, slow generation times, long lives, often very complex genomes with genetic redundancy, many copies of particular gene families. They're obviously diploid, sometimes polyploid genetics. Tissue culture is a struggle. Decades of work have given us access into some systems, but it's not universal. There's often complex tissue morphology, and of course, plant evolution has put us in the position now where higher plants protect their young. All of the early important stages of development are occluded by maternal tissues and are difficult to get access to. So what we've done is we and others have turned to lower plants as an antidote to this. So this is quite an interesting old diagram. This is from the 50s, a textbook in the 50s, and it's a speculation on the potential form of early terrestrial plants. And you can see here, the individual cells here are marked here, so this is a relatively small scale structure. And we're talking about small terrestrial organisms that might have evolved from freshwater algae that would have, green obviously, photosynthetically green, and they have this flattened prostrate structure in this speculation, and these structures at the top here are the reproductive organs. These are the spore-bearing organs. And the key thing is that these are projected to be haploid organisms with very simple reproduction and even simpler anatomy. And there are existent plant species, the liverworts, for example, which bear some similarity to this projection, if you like. This shows an image of Marcantia polymorpha, which is the best known of the liverworts. It's a weed. You'll find it outside in various nooks and crannies, often growing in cracks in pavements. It has a flattened prostrate growth mode. It's more complicated than the simple model that I showed, but it has many of the features which make it really quite interesting as an experimental model for engineering. I'll show you what all these different features are as we go through, but one of the key features of the lower plants is their ability to propagate. And they're still around. They evolved something like 500 million years ago, and they're still with us. They have this ability to, in the case of Marcantia, to reproduce both by spores, by sexual means, and by vegetative reproduction. So these flattened structures can be induced, for example, under far-red light to make the sexual form of the life cycle. So they're haploid. They have an XY chromosome system, so they're either male or female. And here we see the top surface of a female plant, which has been induced with far-red light, So it induces this change, this phase change. And you see the literally eruption or emergence of these features from the flattened structure. This is the top prostrate surface of the flattened sort of a giant leaf, if you like. And you can see emerging from the top structure, undergoing morphogenesis as it goes, these structures which will make these small palm tree-like structures, for want of a better word. So this shows an image of a female and a male spore-bearing organs. So the plants are down here out of focus. So under fire red light, like in late summer, they'll make these structures. And the males make small spore or sperm-bearing structures on the top surface. And with a water drop, those sperm will be then expelled and can be transferred by water splash or by other means to the female plants who might be nearby. And the female plants have this spoke-like arrangement. Underneath each of those spokes, there are archegonia, or the female receptacles, and transfer of these motile sperm and chemotaxis results in fertilization. So if you take a single male and a single female plant and leave it for a couple of weeks, you end up with this. So this is now a fertile, fertilized female plant, and you can make out these small yellow sporangia. And these are spore-bearing vessels which might have a couple 100,000 spores inside each one. So a single cross will produce millions of progeny. And the progeny are in the form of these single cells. So about the size of a yeast cell. They're very resistant. You can store them indefinitely at -80. In the dry, in the room temperature, they'll last about 12 months. But they are these hardened spore-like structures. They, in turn, can be germinated. So if you take them and put them on an agar surface, for example, after sterilization, you can see after about 1.5 days the emergence and enlargement of the cell to make the spherical cell, and you can see now that the chloroplasts have emerged. So you can continue that process. After 2.5 days, you see the first cell division taking place in this germinating spore, and here's an ungerminated spore for reference. So after 2.5 days, all in the open under the microscope, you can visualize these direct processes of differentiation at the subcellular level, and as time goes on, at the supercellular level, we see here differentiation of the first root-like cell or rhizoid, which Liam Dolan in Plant Sciences is notably working on. And you see cells at the top, the apex of the this germinating sporling continue to proliferate and differentiate. And as it goes forward, you start to form this flattened structure as it emerges, and then forms after 5 days on an agar plate, or 7 days, maybe depending on how fast they're growing. You can see the formation of this flattened structure. You may be able to make out the root-like cells diving into the media. So you get the formation of these small elements which are to give you an indication of scale. They're like colonies, but they're plants. They're whole plants, whole organisms, which have grown in place over the week, and there's a pet tip for scale. So it means you can start to think in very different ways of working with a plant system, and there are a whole series of advantages compared to higher plants, and I've given you a laundry list here. Probably the things to point out are that you've got haplogenetics, so if you're used to working with coli, it's familiar. You've got this very fast generation time. The ability to proliferate and to amplify tissues is extraordinary. The other thing to note about the lower plants is that the regeneration of the plants is a default. You don't require external hormones like you do with higher plants. The default is that they will grow and repair themselves. And I'll show you some more direct images of that later on. And you've got very simple morphology, which is directly accessible to all modern high-resolution forms of microscopy and quantitative imaging, for example. And there are other features there that are shared with higher plants, like the basic core of gene regulators and things like chloroplast architecture are highly conserved across the higher plants. And we've got a genome which is about 280 megabases in size. There are 8 autosomes and a sex chromosome. This is a diagram from derived from TAC1, which is now the global type strain from Arcantia, and that was driven by John Bowman, Taki Kochai, and others in Melbourne and Kyoto, including ourselves. It's like a group effort, a community effort to to sequence and annotate the Mercantia genome. We've also looked at a couple of Cambridge isolates, which have slightly different properties, which we think are better for engineering. So we have a genome for that. And in our attempts, so you're used to genome databases, right? You look at a genome database and it's got the contigs and the chromosomal material spread out. Most genome databases are designed from a geneticist point of view, trying to identify a gene, for example. We wanted a database from an engineer's perspective. We wanted to be able to create something which had and here we've a schematic version of the kind of large chunks of DNA that reside in genomic databases. But with those kind of databases, you can use transcriptomics to map transcripts on the genome. You can use bioinformatics to identify and look to confirm the identity of potential coding sequences in those messages. And you can potentially identify putative functional domains that might correspond to genes in those chromosomes. And of course, that's the major effort in most genome databases. But what we've also done is to take this kind of database, we have one called MahoDB, which is designed to take the genes out of context and present them as functional units, both as a source of new parts and as a way of interrogating gene activity inside those processes. So this is a summary of what MahoDB is. It's essentially, it revolves around all of the discovery efforts of sequencing and transcriptomics and annotation, etc. But then it presents the sequence as a series of genetic elements which are then divided up into arbitrary parts based on bioinformatic and comparative analysis. So it gives us a way of both surveying the genome, but also automatically outputting and formatting sequences as parts that can then be synthesized and used in engineering experiments. So it's a way of really trying to present the genome and to deconstruct it for engineering purposes. So that's all well and good. Okay, you can make parts, which are obviously nucleic acid sequences of defined function, but of course you want to use them. You want to put them into other larger circuits. And in our case, we're part of a larger community, we want to share it. So as part of this, we've also put together, this is part of Oakland Park, which is a cross-UK initiative, mainly Cambridge and Norwich, but involves a number of other participants, including people from Oxford. And it was an effort to try and standardize the type 2S assembly part definitions that have been popularized by the success of techniques like Golden Gate, of MoClo, ENSA, of Golden Braid, all of which use the properties of type 2S enzymes to create more efficient ways of putting sequences together. But as time has gone on, small inconsistencies have emerged in what was a unified front when the first time it was published. But as new people came and adopted it, small inconsistencies started appearing. So this is a community-led effort to try and say, at least for plants, this is the recommended way of working. And to make that more concrete, so this is the way that with this Phytobrick or common syntax for plant systems, you take what is at the top here, shows a schematic view of an arbitrary plant gene, where you've got transcribe region, coding sequences, 5' and 3' UTRs, and of course introns and things like that. That they can be divided and separated into modules where these 4 base pair overhangs, represented by these clusters of 4 base pairs, correspond to the overhangs that are used for assembly. So if you're building a whole gene out of a number of different parts and you have a choice of which parts you want to use, you stick to those conventions and you then have something which is both highly compatible with existing processes but completely compatible within this collective plant community. And we thought that was quite important as time is going on, and not just to have a physical standard, but also a legal framework for sharing them. It's all very well to have in your own lab to have things convenient, but the whole point of this is to build a larger commons, a way of distributing and sharing materials for the nuts and bolts aspects of synthetic biology. And this is illustrated here. So this is a part of a collaboration between Oakland Park and the BioBricks Foundation based at Stanford. And Linda Carl in the BioBricks Foundation, who's a lawyer and a biotechnologist, has drafted this open MTA. So it's a materials transfer agreement which is designed to put material into the public domain. So it allows people not just to share things freely between academics, but also to allow people, after sharing, to then subsequently share those materials, just to have this viral aspect of sharing material, and to also share them with commercial prospects. So to use these, potentially use these nuts and bolts aspects for commercial purposes as well. So this is part of an attempt to introduce triage into the kind of work that we normally do, where many of the things that at the moment are distributed by UBMTAs, which is the standard in the field, which allows you to distribute things from one group, one academic group to another, but not to share it beyond that, and not to use it for commercial purposes. We think there's a need underneath this, things which we know are just nuts and bolts stuff. They're the things that we use every day, the kind of things that might have fallen out of patent in higher plants, but in new systems, we just want to make sure that they're accessible and to benefit from the mutual use and sharing of that information and to build a sort of social network around that effectively. So that's the mechanism that we're now trying to negotiate with people like Adgene for distribution of materials of these standardized parts. And of course, these standardized parts are based on this sort of revolution in DNA assembly that's happened recently, where we've got multiplex assembly and highly efficient ligation because of the use of these type 2S enzymes. And the key thing, of course, is that when you take these type 2S enzyme assembly protocols, the key issue is that you don't need to isolate fragments, just puddle the plasmids together. The cleavage reactions happen inside the ligation reaction, and the enzymes are present there because the appropriate products, the correct products, do not have the sites that you use to liberate the fragments in the first place. You're creating something which is immune to the enzyme. So that both allows you not to worry about fragment purification, which is great, but it also means that it pushes the reaction forward because any illegitimate products that might statistically come together are then subsequently recleaved. So it pushes the reactions to completion. So they're both easier and highly efficient. And of course, this is, in my estimation, this has kind of won the battle in terms of, at least up until this point, for best DNA construction in synthetic biology, where it's not so good for genome scale assembly because you've still got scars, but in terms of routine gene assembly, we're seeing this now, the successor, if you like. So this has been popularized by Golden Gate and Moklo, those of you know familiar with the field. This shows from Adgene the, I think it's 95 plasmids that are required to set up the Moklo system. So because you can take genes and assemble them as pieces and then larger and larger pieces, you need this nested way of working to bring things together to make these larger constructs at the end of the process. So you just start from a broad base, like a pyramid, start from a broad base and work towards your construct. So as part of the work in Cambridge, what we've been looking at is trying to simplify that. And so we've adopted this recursive way of working, where we have the same kind of principle of type 2S assembly, but now instead of going from the base of the pyramid to the tip, we have this recursive way of working. So we start out by using our standard parts, the same as everyone else uses and now standardized. You can assemble them using a single enzyme commonly used, BSA1, which is the common used enzyme for level 0 parts. And you can assemble those into transcription units in 1 or 4 different plasmids, which can in turn be assembled using a different enzyme, because these are specific vectors designed for this. So using SAP1, you can assemble those into so 4 of those can go into one of these plasmids. So you can amplify them 4-fold at each cycle. So you can go back and forth. You have 2 sets of 4 plasmids, and you just amplify 4-fold as you go back and forth through each one. To make it a little clearer, it's shown here. So you would start with a level 0 part, put these together, you choose the parts that you want to make for each transcription factor, which is 4 sets of each one. You put them into one of each of these 4 plasmids, and then you've got these 1 transcription unit per plasmid, and then you can do the same thing again. If you have 4 of these, you put it into one of each of the 4 receivers, and you've got an odd and even set of plasmids, and you alternate between the 2 type 2S enzymes, and you go from individual parts, which you keep in the freezer, to 1 transcription unit, to 4 transcription units, to up to 16 transcription units, or more if you can cope per plasmid. So it's a relatively simple way of working. Lowers the number of plasmids you use Same principle as Golden Gate and MoClo, but it's much easier to keep in your head because you don't have this huge number of plasmids involved. And so this is just an example of some of the things you can do with it. So we've made a series of, so we use it, call it loop assembly, because it loops back and forth between the two enzymes. And this shows one of the level 2 loop assembly vectors. So again, adapting it for plant work, we have an agrobacterium compatible plasmid backbone, which goes for coli and for agrobacterium, with left and right borders for plant transformation, and then slots in here for 4 different genes in this level 2 plasmid. So if you go to level 3, you've got 16, but here you've got 4. There's an antibiotic persistence marker and 3 different color fluorescent proteins with slots for different promoters in this case. And of course, it could be anything, but you basically have this conceptually easy idea that you've got 4 or 16 slots that you can play with, and you can reorder them very easily just by, depending on which plasmids you put together in the reactions. And of course, one of the big advantages of the type 2S assemblies is that you can bring automation very easily to the piece, and we're using lab-side echoes for nanoliter scale assemblies in collaboration with Nicola Patron in Norwich. But by putting these things together, you can make different constructs. And here we have a young propagule of Marcantia showing different color fluorescence with 3 different promoters working inside the organism. And this is where I want to introduce this propagule is rather curious. I mean, I haven't introduced this yet, because on top of the Marcantia plant, you don't just get the sex organs, you also find these what are called splash cups in common parlance. And you have, for example, this cup-like structure, and then there's a cavity underneath that. And you can see inside that cavity, there are these small clustered objects, which we were just looking at. You can see a few of them being splashed up here. These are called gemma, which are the asexual propagules of the plant. So as you, say, make a transgenic plant, as it grows, it will spontaneously make these splash cups and start producing these clonal propagules. They're derived from single cells from an immunogenic layer at the base of that subtending cavity, and they just accumulate in there. They grow to a certain size and then stop. When you, however, take them out, you've got this very nice discrete architecture, and when you take them out, you can grow them. So if you put them onto agar, for example, they go through this developmental sequence, ultimately making more splash cups. So it's very easy to propagate them after a few weeks. And the relationship between the cells within the gemma are very easy to follow, because all this is happening in the open. So you can visualize growth at cellular scale directly under the microscope. And to make that a little more clearer, this is the gemma that's essentially an overnight imaging growing after the process of transfer to agar. And we're looking here at nuclear localized green fluorescent proteins and membrane markers to outline the cell under a confocal fluorescence microscope. So you can visualize directly what's going on. You can, for example, this is on day 2 and day 3 after those gemma have been transferred to nutrient agar in a plate. And you can see how much they've grown in a 24-hour period. And under the confocal microscope, you have high resolution access to that top surface. You can see, of course, the individual cells here. So, in the right direction here. Where is it? So the cells, you can make out individual cells in that picture. If you take those 2 images, one at day 2 and day 3, what you can do is to use, for example, warp registration algorithms. So you can take those 2 images and now warp the day 2 image so that it looks like itself a day later, by simply warping and stretching to match the salient features of the day 3 image. And I show them in different channels here. So you've got those 2 images, which are now geometrically superimposable, which when you do that, and you can, a bit bright in here, but you can make out, I hope, that you can see both the outline of the cells, and in yellow or yellow type color, which is where the same cell walls or outlines are present in both day 2 and day 3. Any feature which is appearing between those 2 time stages, for example, at day 3, shows up as a red feature. So for example, you can see in here around the notches in the gemma, where all the cell division takes place in these 2 meristematic notches, you can see lots of new cell walls. So this shows the number of cell division. Each one of those red lines corresponds to a single new cell wall that occurred in that 24-hour period. And the cell index here is, so it's like 20 hours or so is the cycle time in these rapidly growing plants. So you can measure those, and I think there's an image here which shows in more detail the arrangement of these new cell divisions. So you can map processes like cell division directly onto the tissue. Of course, other features like rates of growth or cell expansion can also be mapped. So here we've got another gemma. This is using this process of taking a time-lapse sequence and from one image to the next, looking at pixel displacements. So you can get a quantitative map of growth, and you can see it's pulsatile across the growing gemma. I think it's over 2 days, if I remember correctly. And this is work by Mihail who's been looking at these, ably assisted in the early characterization by Neri Perswani. And so you can visualize and quantify these processes quite clearly. And so what Mihail's has done is to take these even further forward, because here we're looking at, there's two forms of gemma here, and you might make out that on the left-hand side, we've got a gemma and we've highlighted that. On the right-hand side, it looks weird because it's been surgically cut in half, or into thirds in fact. And so the middle portion of the gemma and one of the apical portions is separately sitting on agar as separate pieces. So if we look at the wild type, so normally you get division at the ends, these little notch-like arrangements, and they act as meristems or growing points. There's also cell division around those. And in the middle portion where you see that orange box, there's not much cell division, there's really no cell division in that middle period, middle zone. So what Mihail's was doing was then taking cells or tissues out of context by doing microsurgery. And if you now look at a time lapse of this one, you can see now that the cells in the middle of what was the middle of the gemma are now dividing rapidly. So this onset of division, which is very different to the normal context. In fact, it's much more like you see in the apical zone of the meristem. So he's pursued this, and he has, for example, here there's a version of the same experiment, now with a marker. It's at the top here, it's the YACA2 promoter up the top here. YACA2 is a gene that's involved in the last step of auxin biosynthesis in plants. There's a hormone that's involved in polarity and growth. And so if we kick off this image, so first of all, we're seeing that the emergence of expression, high levels of expression of the promoter driving now grid fluorescent protein. So we're seeing genes that are normally associated with auxin expression now turning on in the middle portion of the gemma. We know that auxin in Marcantia is associated with dorsoventral polarity. So this shows, for example, one of the normal gemma, if you like, with, you can see these hair-like cells, which are these gemma, which in the case of addition of here 10 micromole of an auxin, naphthalene acetic acid, you see now that one of the gemma is much smaller, and it's festooned with these hairs, which normally only occur on the ventral side of the organism. Don't forget that these gemma are made in these flash cups, and they're symmetric when they start out life in the splash cup, but when they're expelled from the splash cup and land on a substrate, then they have to very rapidly adopt a particular polarity, because the hairs, the root hair-like structures, are required to mine the substrate. So they actually have precursors on both sides of the gemma, and when they land after and germinate, only one side of that gemma undergoes the large-scale proliferation of gemma, and that's believed or now shown to require auxin. So there's a flow of auxin which is associated, probably gravitropic and light regulated, which drives this creation of polarity inside the systems. And here we've now got the application of auxin. So before we were looking at the rates of cell division and the role of auxin synthesis genes in that process. So here we have the normal process of growth where the middle zone is not affected. And here we have now in the presence of auxin, I hope it's not too dark, we'll go down the bottom. But so you've got now, in the presence of auxin, you've got growth, the cell division is suppressed. I hope you can see that in that. Go back. So that's the wild type. In the middle portion of the gemma, there's not much cell division. There's lots around the tips. And now looking at, in the presence of auxin, you see lots of cell expansion going on, driving growth, but not much cell division. And this is now looking at the separated, not just the intact gemma, but separated ones. This is the control over this side on the left. So you can see that when you separate them, you've got this burst of cell division in the middle portion of the gemma, which normally doesn't happen. But now we expose it to auxin, and you can probably make out that I'll run that one again, because as you watch that, it's looping good. So you see the emergence of these rhizoids from the top surface, and you see them growing out very quickly, responding to the auxin. And you also note that the cell division has been suppressed in that middle portion of the gemma, whereas in the apical zone, it's behaving normally. Well, it's suppressing division in the notch, but still expanding. So we're developing this view that there's, in this very simple test bed, you've got the emergence, or the role of auxin at least, in suppressing that middle zone of the gemma. And we think that's related to auxin production and response. And it's clearly just the start of new ways of working. What's very interesting about the Marcantia system, and this is shown in a review from Dolph-Weijers and Doris Wagner, what they've shown is that if you look in different model organisms, so here we've got Arabidopsis, rice, Fiscomatrella the moss, Marcantia, our favourite liverwort down here, and now green algae down here. What they've done is they've taken arguably 3 of the most important classes of genes associated with auxin response, and then looked at the number of gene families. So in Arabidopsis, you've got half a dozen different auxin response elements, tier proteins. You've got 30 and 25 different AuxIA or ARF genes. Even in something which is relatively compact like Arabidopsis, you've got multiple gene families. And as you go down through the list of plants here, down to Marcantia, you see that there's a single auxin response gene. You see there's a single transducer, and there are 3 different response factors. And it turns out that there are 3 different classes of response factor of these ARF genes. So we've got this extremely streamlined, for whatever reason, streamlined genetic system, which is causing the response to auxin, triggering the response to auxin in Marcantia. So in terms, and this is found across the board in Marcantia. So if you look here, you've got, from the Genome paper, you've got a picture of Marcantia shown in that purple set of figures in the middle of this diagram, looking at the number of genes in the bottom line here on the x-axis, gene families that are found across terrestrial plants, running from green algae right through to Arabidopsis as a higher plant representative. And you can see at this end of the scale, you've got large gene families, the MIB genes, the basic helix proteins, RAS genes, etc. And in the case of the higher plants, you've got these enlarged families of genes, in some cases up to hundreds of genes in these particular classes, whereas in the case of Marcantia, they're highly reduced sets. So from an engineering perspective, that's attractive. We're now using the simplicity and the streamlined nature of genes. There's less than 400 transcription factors in Marcantia at all. And we're trying to make markers for essentially to illuminate the behavior of these processes inside Marcantia by making parts that correspond to, for example, promoter domains for the whole comprehensive set of transcription factors, and using the MahoDB as a way of automatically exporting the different regulatory genes and implementing using loop assembly vectors to make these multi-spectral quantitative markers. And to make that a little more clearer, so this is just now focusing on the meristematic notch of Marcantia in the early gemma. And you can see here there's a couple of key genes, Baileyini meristem, scarecrow, shoot meristemus, and a control showing broad expression. So we're looking and trying to use these as switches or markers that we can get a comprehensive set and share those through these standardized ways of working. We think that here we have in Marcantia a test bed that we can use to create this sort of universal tool of parts, not just for probing, but also for measuring what's going on inside the tissues. And if you think back to the whole plant, I just want to end with a small section on what our long-term goal is. And this shows a section across an adult Marcantia. This is a portion of the meristematic notch in an adult plant, and this is a splash cup. Now, if you look at that image, you'll see that the top surface, aside from the features that I just pointed out, have this sort of modular, almost like jigsaw-like arrangement, repeating arrangement. And they're marked by those donut-like structures there, and there are modules that repeat across the top surface of the plant. So in detail, look more like this, where you've got those donut-like structures turn out to be air pores. They're sitting over the top of air chambers, and they form in a modular fashion. Essentially, as you watch these things grow, all of the time-lapse images I showed you, if you take them and extend them out after 5 days or so, they'll start assembling these structures on the top surface of those young gemma or spawlings. And these particular repeating domains are an example of a very interesting three-dimensional sort of morphological structure, if you like, where this is an image from Leopold Knee, so it's probably rather more simplified than what reality is, but it shows the key salient features. You have these chambers which from the top view look like donut-like structures made up of 16 cells, which act like a chimney effectively, and on this chamber, this air chamber that you see underneath it, has a roof, has walls, has a floor, sits on top of storage tissues underneath, and inside it has all these filamentous, highly photosynthetically active cells inside the air chamber. So this is, in something like a Marcantia, is the equivalent of a leaf. It's a module essentially optimized and designed for photosynthesis. And so that is dispensable. You can use CRISPR-Cas9 techniques. So here we've got a guide sequence which is targeted at NOT1, which is a gene that Taki Kochai's lab has shown is required for air chamber formation. We have Marcantia lines that have the Cas9 gene embedded in the genome. So all you have to do is deliver the one or more guide sequences, and you get very efficient gene knockout. And so using this, you can see from the wild type, you shift towards these defects, which have effectively a much more transparent appearance, because the entire top surface of the gemma or the thallus is missing these air chambers. So these air chambers under lab conditions are completely dispensable for growth. So you have a very interesting genetic target that you can then know. You have a target that can be completely be dealt with in a facile way. And so we're looking now at developing markers, and the processes of formation of these air chambers starts very close to the meristematic notch. So this shows the meristematic notch is in here. It's a single cell, is the apex cell in the middle of that notch. And you can start to see here markers which are showing particular patterns, which probably show up better on the next slide. So you can see particular markers here, where you've got fields of cells which are undergoing proliferation and patterning to create the formative divisions that will allow these air chambers to form. And so you can use confocal microscopy to get images of these key. It doesn't show up very well in this diagram, but there are key recognizable features. Perhaps shown better. This is a very nice paper from Apostolakis back in the 80s, who used light microscopy and EM to look at some of these formative divisions. And you have these characteristic emergence of spaces, like schistogenous divisions, and formation of these characteristic square division planes, which are characteristic of the initiation of this early stage of air pore development. And building on top of that, markers that will mark, for example, using the same kind of promoter strategy, things that are either inside the air chamber or like epidermal here, or the filamentous cells here, and creating markers for what's going on inside this chamber. We think this is a really nice target to start to address some of the questions that I started at the beginning of the lecture, talking in more general terms about the formation of the picture of a pitcher plant, where of course you've got many millions of cells involved. But here you have a small handful of cells also making a defined, recognizable three-dimensional structure with particular key cell types, and which is much more directly accessible than any of the higher plant systems. And one of the side benefit here is that one of the other interests in the lab is not just the structure itself, but what goes on inside it, in the sense that we have chloroplasts that are doing all the work, and I think it's hard to escape chloroplasts in plants. Basically, plants are self-assembling bioreactors for chloroplasts. All the hard work of bioproduction and photosynthesis is done in the microbe-like compartments. It's basically a self-assembling bioreactor for refactored microbes, which are chloroplasts. And they have tiny genomes, of course. So there's 120,000 base pairs in the Marcantia genome. It's highly homologous with all higher plant genomes, some recombination, but they're effectively the same set of genes with a few exceptions. And they are capable of huge levels of bioproduction, huge levels of protein expression, and of course are the source of most ATP and reducing power inside the plant, and a home to many biosynthetic pathways. And you may be aware that one of the main bottlenecks for working with chloroplasts is the transformation and homoplasty aspect. So we've taken a slightly different approach to that, and I've just, the last fleeting slide. So we've got, in the case of Marcantia, you've got these wild type cells with their 30 or so chloroplasts per cell. This is a mutant, a CRISPR-generated mutant in Marcantia, where we've knocked out, we've targeted a few genes which are known to be required for chloroplast division, and which in Arabidopsis have been well explored by people like Kevin Pike for making large chloroplasts. So this is a giant chloroplast phenotype reiterated in Marcantia, where we have relatively few and larger plastids per cell. And you may be aware that hornworts, which are a sort of sister primitive plant clade, have 1 or 2 chloroplasts per cell, so it's not that unusual. And the key thing here is that you've got inside each chloroplast genome, you may have 50 to 100 copies of the genome inside each chloroplast. So when you do a transformation experiment, you not only have to get your DNA in, you have to then do essentially a population selection to allow it to proliferate through that plastid and through that cell and through that organism. So that's quite difficult. So here we have at least trying to minimize the population pools there. So just to finish, we've tried to use Marcantia in this integrated way, trying to knit together cellularity and DNA standards and ways of working, and to use that as a test bed for the kind of supercellular aspects. And initially one might think that multicellular systems are going to be more difficult to work with than, say, microbes. But if you think about engineering a pathway in a microbe, where you might have 5,000 genes, 12,000 genes, that you're working inside a pool with that number of interacting elements. And in a plant context, if you use cellularity as an abstraction, you can use cells essentially as black boxes and use cell differentiation as a way of creating essentially biological factories in a cellular context rather than a genetic context. And so just to summarize that, we've got these different elements of trying to build a community for plant work around these lower plants. And that has led on to things which are underway at the moment, looking, for example, at cell-free systems, which can be freeze-dried and non-GMO for teaching contexts. And we're doing quite a lot of work with the Global Challenges Research Fund mediated work. And those of you who are maybe interested in the collision between biology and the physical sciences and engineering, we've been using instrumentation, bioinstrumentation, as a way of bringing people together. We have this biomaker.org site and Biomaker Challenge, which runs every summer. And if anyone's interested in sort of building bridges between Cambridge and Oxford around something like this, really love to talk, because there's lots of opportunity for doing that. And I'll just finish with there's lots of folk who've been involved with the work, both in my lab and others. And here we've got folk in green are working on plant systems and cell-free and bacterial systems shown in black. And these are alumni, people who've moved on to bigger and better things, and the current members in the lab, and lots of collaborations across the UK and beyond. And thanks very much for your attention.
Thank you very, very much for that. That was absolutely fascinating. I could certainly learn a lot. showed melting out these air cores. If there was some aspect of morphology that you would tweak rather than just eliminate, what do you think would be most appealing?
Well, there's been a couple of sets of experiments over the last few years which have kind of set our direction at least. So Dominic Loke in Emeryville has made experiments where he's taken an existing genetic pathway where you have a master controller, a regulator, transcription factor, and it regulates the family of genes, network of genes, and what he's done is he's followed that pathway, taken a promoter from one of the downstream genes, and then used that to cross-regulate the master regulator. So if you then put that hybrid gene back into a transgenic, so the normal process of whatever triggers that process happens, but when it does, the transcription factor, the regulator, the master regulator, feeds back on itself because you've now got a copy where one of the target genes that it controls is now itself. So you introduce this sort of feed-forward loop into the process. Very simple principle, and what Dominica's lab has been able to do is to use that for creating larger cell wall masses by increasing the secondary wall deposition because they've got a master regulator that's responsible for that process. He has master regulators that are responsible for secondary products. So you can then increase the level of secondary product by that. A couple of months ago, Zachary Lippman at Cold Spring Harbor did a sort of complementary experiment where he was working with tomato systems, and he took two genes which are known to be required for meristem bulk and maintenance and initiation in high plants, chip meristemus and bushel, the two homing domain protein genes known to be involved in meristem size and maintenance, etc. So he made a whole stack of guide sequences targeted against the promoters of bushel and chip meristemus, the sort of 5 kb or so upstream of that, and shoved that battery of guide sequences into his plants, generated misconstrued or misarranged, deranged promoters for those two genes, and then just let it happen. So you just then screen what happens afterwards. And he found a whole range of different defects because effectively you're changing the response of the promoters to whatever else is controlling things. So you don't have to know very much. But then phenotypically you find tomatoes with more locules, less locules, different leaf shapes, different leaf arrangements. So if you can choose regulators and change the internal logic, I think those two experiments show that it's working with the system, if you like, using abstractions to, if not consciously or rationally engineer, to at least generate the startings of that, I think. And so in our case, in Marcantia, we're very keen now to extend the libraries that we've been building, which are comprehensive markers for all of the transcription factors in the organism, and to use CRISPR techniques also to identify key transcription factors in particular processes, because they're not well known at the moment. But it should be relatively easy to do because you've got the advantages of being haploid and the advantages of phenotypes are so easy to quantify. So I think there's I think we're due now in the plant systems for a whole renaissance of new types of experiments based on the last 10, 20 years of genomics and developmental biology. And I think that Zach Lippman's experiment is a great example of how that information can be fed into different ways of working. And a lot of the new energy comes from the ability to edit directly in place. So the kind of things he's making in his lab can go directly into breeding programs. It's quite a different paradigm.
So I've got quite a naive question being not a plant biologist, but given the way you get the tissue development constricted by the cell wall, with daughter cells producing a cell wall in between, how much of kind of plant development has apoptosis as part of it?
Very little. There are some examples every fall we see it as the leaves fall off. There are some examples where you've got programmed death, but you know it's the rule rather than the exception in animal systems. You use musical chairs type arrangement where everyone proliferates and the signals come and go, and then if you're not don't find your partner, you're gone. In plant systems, it's marked by plasticity. So you've got proliferation, which itself is quite highly regulated, and if, for example, you do ablation experiments, laser, surgical, or whatever, things adapt in place. So the local interactions between cells are dictated not just by secreted molecules, but also cytoplasmic bridges. So, for example, there's a whole range of different ways of communicating locally in plant systems, but there are things which don't exist in animal systems. So transcription factors that might have a role in laying down a piece of architecture in a tissue context often can move between cells through plasmidesmata, which are the equivalent of gap junctions, but of course you've got a cell wall there, so they're actually physical pathways, and they're regulated. Most viruses in plants move through plasmidesmata. They don't bud through plasma membranes, they move. And there's this whole conception of a symplast. So older cells tend to be more isolated, but in young tissues like meristems, everything's connected, but it's regulated. So you've got more of a colonial view of things. Everything's connected. You know who your neighbours are, and your properties are part of that community.
Okay, that's great. And given that you've got such exquisite maps of the cell divisions that go on in those early gemma, are you seeing cell division in exactly the same place in different planes? Is it that well programmed? Or?
I think the local adjustment to conditions is such that it's like when you do in a surgical experiment, you can make a change if it's genetic or physical surgery. A change will then propagate through because everything's connected. So, for example, we did some experiments in Arabidopsis years ago where we had a diphtheria toxin being triggered, and we were using systems which have very precise patterns of expression. So you'd expect in a static system that those cells would be gone. But of course, they're not static. They're built, like the pitcher plant, it's built on the fly. And so when genes are turned on in order to build something, then of course if they die, then they lose their, undergo death. But then cells around them are trying to take over that fate because it's all part of a self-repairing system. So then they then turn on this novel toxin. So you get these cascades of artificial cell death. And similarly with markers, you can generate systems which can be put in place which in the wild type context are very precise, very controlled. But when you put something out of balance, where the system is trying to reset itself, you can then get secondary processes that then proliferate and propagate through the system. So that's actually paraphrasing one of the big challenges, I think, which is to understand better what the global relationships are, what's the order of signaling and interaction that's required to go from step to step to step. And that's why I think the air chambers are quite a good target, and for two reasons really. One is that epidermal features tend to be easier for genetic analysis because they're supernumerary. It doesn't matter if you make all these mutants, they don't die or anything. They're happy, effectively, so you can analyze them easily. And secondly, the processes, certainly in higher plants, where you're looking at guard cells or trichomes, big sort of hair cells, or root hairs, they've been well examined by these kind of genetic techniques and shown often that you've got isolated pathways and you can reconstruct the steps. And thinking of the kind of rejigging or reprogramming of pathways for reprogramming development, you need that kind of map. And it needs to be in the context of the anatomical features at the cellular scale as well. So that's the sort of challenge.
I'm just wondering if you've used similar techniques to try and understand differences in cell differentiation and gene expression in forming female versus male gametes.
Yeah, we haven't actually tackled that one, partly because we're choosing to work with the lower plants because all this is exposed development. And a lot of the gametogenesis is actually buried inside tissues. Not greatly, so it's just a few cell layers down, but it makes it difficult to use high resolution microscopy. Whereas, the images I've shown you, like the dorsal surface of the gemma, for example, there's a sort of a deliberate intention there, which is to look at things that are easy. It's like looking under the light for your keys, right? But in a way, but it means that you can do a lot of things in a more constrained domain, which if you wanted to look albeit very interesting things like sex determination or the diploid part of the life cycle, it's more difficult to do. Not impossible, because there's lots of clearing techniques and other approaches, but we've tried to do the easy stuff.
Does anybody have any other questions?
Just one, maybe naive one. But I was thinking about your method of adding insets and passing it. What is the maximum number of insets on
Bernardo and Fernando Federici, who set the system up, have routinely got to 16 constructs. Beyond that, you can go up to 64 theoretically, and also you can use spaces, so you don't have to have the set of 4 multiples of 4. One of the things I didn't mention was that because it's recursive, it goes back and forth, you can skip a loop. So if, for example, you want to make a synthetic promoter with lots of little domains, you can use them like 4 or 16 at a time, and then introduce them back into an existing construct, like a normal level 0 card. So if you wanted to make a gene with a highly repeated domain in the promoter region, for example, you can loop through, get your little oligos or whatever lined up, and then plug it back into a normal assembly. And so that's increased the number as well. But 16 is about right. 4 is probably routine. Great, well, thanks again.
Thank you very, very much again.